Welcome to the Shark Tank. Entrepreneur Jake seems to know a lot about pharmaceuticals, but will his new medicine be enough to calm the feisty sharks, or will his dainty drug prompt some unwanted side effects with them instead?
Hey sharks, have you ever had a urinary tract infection, or pneumonia, bacteremia, or any kind of respiratory infection, especially those associated with cystic fibrosis, osteomyelitis, or diabetic food soft tissue issues?
No, I don't think so.
Not that I'm aware of.
Oh, I actually came down with a urinary tract infection this morning.
My company is called Tobramycin Takeoff, or TT for short. I have invented a new drug called Tobramycin that can treat all of the infections listed. We currently have 4 routes of administration for the drug: injection, inhalation, EV, and ophthalmic. I am asking for $100 million for 10% equity in my company.
Very interesting product. But have you made any sales?
Tobramycin had North America sales of roughly $75 million last year. Although sales have grown in the past 8 years, I have to be honest with you guys, they're expected to stabilize moving forward.
Yeah, that's all great, and I definitely appreciate your honesty, but now tell me what I really want to hear. What are the key functional groups of this drug?
The key functional groups in this molecule are amine, ether, and hydroxyl groups. There are 5 amine groups, 4 ethyl groups, and 5 hydroxyl groups. The 2 types of non-covalent bonds that could occur with these functional groups are hydrogen bonds and hydrophobic interactions.
Alright, now look, all this is great, but how does the drug actually work? What specifically is the drug target? Are there any non-covalent interactions? And what's the mode of inhibition? If you can figure all this out for me, I might be able to cut you a deal.
Tobramycin is comprised of hydroxyl and amide constituents. They are surrounded by negative phosphate groups, pictured above in blue, in the heart of the protein molecule, resulting in many hydrogen bond interactions. Looking at the protein drug complex as a whole, the hydroxyl groups are once again congregated near the blue phosphate coils. The ligand shows with a 3D surface, as pictured above. Once again, it is surrounded by the hydrophilic structures, pictured in blue. The 2 key types of non-covalent bonds are hydrogen bonds and hydrophobic interactions. The bulging conformation of 2 adenines are stabilized within the binding pocket by the hydrogen bonds between phosphate oxygens, water molecules, and exocyclic atoms of the invariant amine portion of the aminoglycosides. As for hydrophobic contact, the positions of the binding sites are in hydrophobic cavities of the subdomains IIA and IIIA. The hydrophobic residues that come in contact are Val-293, Pro-339, and Asp-451. Thus, hydrogen bonding and hydrophobic contact result in tobramycin to bond irreversibly to the aminoglycoside. As for mode of inhibition, tobramycin binds irreversibly to one of 2 aminoglycoside binding sites on the 30S ribosomal subunit, inhibiting bacterial protein synthesis. Tobramycin may also destabilize bacterial membrane by binding to 16S RNA, an active transport mechanism for aminoglycoside uptake is necessary in the bacteria in order to attain a significant intracellular concentration of tobramycin.
I'm completely sold on this. I think it'll really take off with the customers. I'll give you the $100 million, but for 20% of the company.
No, I can't do that. Do you even know the implications of the 30S subunit in disease? Tobramycin is found under the class of drugs known as aminoglycosides. It is a type of antibacterial that competitively inhibits the translations of mRNA into proteins by binding to the active site of the 30S subunit. When tobramycin binds to the ribosome, it causes a conformational change in the active site that prevents the ribosome from reading the mRNA sequence. Therefore, the bacterial cells cannot perform protein translation and cannot grow or replicate, thus killing the cell.
Come on now, Jake. Who do you really want to work with? Not this cheapskate over here. I'll offer you $100 million for 15% of the company. This is really revolutionary, and I could get this off the ground with my revolutionary infomercials.
How do you expect me to make that work? I'm not a charity.
You know what? No. Everyone stop. Jake, I'm in. I'm going to give you the $100 million for 10%.
Well, it appears Jake got the deal he wanted. It should be an easy decision for him.
Well, Tanner, now that all the offers are on the table, the choice is pretty clear to me.
Do we have a deal?
Oh, baby, let's do it, Jake.
Sorry, Tanner. If you want to play in the big leagues, maybe throw in something a little sweet next time.
You're missing out, man. You're missing out.